Secretions into the GI tract are critical and help with the digestive process. Secretory glands throughout the GI tract secrete enzymes to digest nutrients, mucus to lubricate and protect the GI tract, as well as water and electrolytes to make sure that the intestinal contents are fluid while digestion and absorption are occurring. Most secretions are formed in response to food in the lumen, and the amount that's secreted is the amount that's required for digestion to occur. Every day, the GI tract is exposed to a total of about 9 liters of fluid, about 2 liters from ingested food and drinks, and about 7 liters from the secretions of the salivary glands, gastric glands, liver, exocrine pancreas, and small intestine. Of this 9 liters, almost everything is reabsorbed and mostly by the small intestine. Only about 100 milliliters is excreted. This massive absorption in the small intestine is primarily due to the osmotic effects of nutrient absorption. So as nutrients are absorbed in the small intestine, an osmotic gradient is established, and that drives the movement of water. Secretory units have different structures throughout the GI tract and can be single cells, tubular glands, or complex glands. Individual cells within an epithelial layer can produce secretions directly onto the surface of that epithelium. Mucus glands are an example of this type of gland. These cells secrete mucus onto that epithelial surface where it adheres and protects. Tubular glands are invaginations of the epithelium that contain secretory cells. Secretions enter the lumen of the tube and flow up and out into the lumen of the GI tract. Tubular glands can have different arrangements. Simple straight glands can be found in the stomach and the colon. Simple branched tubular glands and simple coiled glands are found in the lower part of the stomach and in the small intestine. Complex glands are those found outside of the intestinal mucosa. Secretory cells are arranged in clusters around a ductal system. The secretory cells secrete their product into the duct, and that duct carries the secretion to the lumen of the GI tract. Sometimes the cells lining the duct here modify the secretion by changing the electrolyte and water composition as it flows through. Then the final secretion is what's emptied into the lumen of the gut. The salivary glands and exocrine pancreas are examples of these complex glands. In this diagram, there's a tubular exocrine gland secreting its product into the lumen of the GI tract. Contained within this epithelial layer, in addition to the exocrine cells, are enteroendocrine cells, and they secrete hormones into the blood that act on distant target sites. Glands in the GI tract are generally stimulated by the presence of food in a particular segment. Part of this is through direct contact stimulation of the surface glandular cells by the food. This is particularly true of mucus secretion by mucus cells that lubricates the GI tract. There's also autonomic stimulation from the parasympathetic and sympathetic nervous system. Parasympathetic stimulation generally increases rates of secretion, especially in the parts of the GI tract that are innervated by the vagus parasympathetic nerves. That's the salivary glands, esophageal glands, gastric glands, pancreas, and Brunner's glands, as well as the distal large intestine. And in contrast, sympathetic stimulation generally reduces secretions by vasoconstriction of blood vessels supplying the glands. There are also several hormones that help to regulate the volume and composition of secretions into the GI tract, and these hormones that regulate GI function are primarily released from the enteroendocrine cells in the epithelium in the presence of the luminal stimuli. The hormones are absorbed into the blood and are carried through the circulation where they then stimulate secretion. This type of stimulation is particularly important to increase secretions in the stomach upon entry of food and to increase secretion of the pancreas when food enters the duodenum. Secretory cells have receptors on their basolateral membranes for a variety of different hormones and neurotransmitters, and different receptors act through different second messenger systems to increase that exocrine secretion into the duct. Now we'll zoom in on a single secretory cell to look at some of the cellular mechanisms that stimulate secretion into the lumen. There are several receptors on the basolateral membrane of these secretory cells, and some receptors are coupled to the activation of adenylyl cyclase. The net effect is an increase in intracellular cyclic AMP, which leads to product secretion. A few examples of signaling molecules that act in this way are vasoactive intestinal peptide, secretin, as well as the presence of cholera toxin in the lumen. Other secretory agents act through this IP3 mechanism. So receptor binding activates phospholipase C, and activated phospholipase frees diacylglycerol and IP3 from the membrane phospholipid PIP2. IP3 causes the release of calcium from intracellular stores in the endoplasmic reticulum, resulting in an increase in intracellular calcium concentration. And that rise in intracellular calcium causes activation of protein kinase C and protein phosphorylation, ultimately resulting in product secretion. A couple of agents acting through this mechanism include acetylcholine and cholecystokinin.